Okay, so chapter 13, we're going to go into evolution. And evolution is, remember, the genetic change when we're talking about the population. So it's not about an individual like Mario here, undergoing evolution. It's the entire population of that species that undergoes evolution. And evolution is done through this process of natural selection. And this is just this process with which the organism will have these beneficial traits within themselves and pass it on to their successive generations. And those traits are beneficial in that environment at that point in time. And so those tend to have those beneficial traits will have a better success rate in surviving and passing on their gene, so reproducing as well. So those are the two big things, to be able to survive and survive long enough to reproduce, to pass on those traits to their future generations. So those with more beneficial traits, like I said, will be able to pass it down to their generation. So like this mantis here, notice how there's a mantis over here. See the little eyes over here and its legs. And this is a good trait to have because for a couple of reasons. One, it prevents predators from finding it for camouflage, and it also helps it to be able to sneak up on prey and be more successful in surviving by the amount of resources that it can take in. And then there's those that are less beneficial traits. And those with less beneficial traits may not survive long enough to be able to pass it down to their next generation. So for instance, you see these different types of moths. Before the Industrial Revolution, cedar trees are kind of actually looks very similar to this white moth. And you notice a little bit over here as well. So at that point in time, when the trees were whiter and spotted in color, which one will be more of a beneficial trait? The white one, right? Because it helps camouflage it, prevents predators from seeing it. And those that have the darker traits here will be picked out by the predators and be eaten up, potentially before it reproduces. And so what happened was after the Industrial Revolution, there's a lot of coal and smoke now building up, and it actually painted these trees with this smoke on it, and so it turns the cedar trees to become darker in color. And at this point in time, now even though they have the same traits and everything, but which one's more beneficial at this point in time? The darker one, right? Because it blends in with the darker trees' trunks now. So, yeah. So there's evidence for evolution, and these are all the different types of evidence that we'll go into. So the very first one will be fossil record. And before we go into fossil record, we gotta first understand what are stratas. Stratas, or stratum singular, are these rock layers that you see even on this picture here. And those that are lower down on this, these rock layers that are lower down here, notice the layers like this. So the ones that are lower down are going to be older or younger. Older, yes, because as new sediment lies on top of it, so the further you go up, the younger things are, because the sediment settles on top of the older sediments. And so those that are lower here will be older, and then the younger ones will be higher up. And so we use the strata that we see within the rock layers to help us to determine how old these fossil records that we find. So the fossil record is just being able to find the fossils within those stratas. So the oldest fossils that we found are prokaryotes, and these kind of follow our the evolution and trend, where the prokaryotes were the ones that are precursor to the eukaryotes. And you don't have to memorize numbers, but those that were found were approximated 3.5 billion years ago. And other things is like transitional fossils help give evidence to evolution as well. And a transitional fossil like this ancient whale shows here are just fossil evidence that actually shows some ancestral species that we can relate to the current species. So although current whales do not have any of these hind limb bones, this ancestral whale has this hind limb bones. So it gives us this evidence that at one point in time, the ancestor of the whales had these bones, which these ancestral whales came from land animals at some time, or at least 4 quadrupeds.
How do they know? How do they estimate that it's been 3.5 billion years? How do they estimate that?
Part of it is through carbon dating. And carbon dating is, we didn't talk about it in the chemistry part, but there's isotopes. Isotopes are, you remember neutrons, right? Within the nucleus. So there are certain isotopes that means that those, just say carbon, okay? We'll just say carbon has 8 protons and 8 neutrons, right? There are some isotopes that has different numbers of neutrons. We still have the same number of 8 protons, but we have a different number of neutrons. And some isotopes are more radioactive than others, and we use the half-life or the decay of that radioactivity to determine the length of time that something has last. So we'll use like the fossils that we find there to see how that radioactive decay, and then estimate a time as to when that fossil originated from, compared to how much decay occurred since from then to now, kind of thing. Biogeography is another type of evidence for evolution, and we use this to find the species' geographical distribution within not only the geography, but also within time as well. So an example is like this bird here. So at some point in time, this ancestral bird was from this area within the Pacific Ocean, and then as later generations occur, then they turn into different species, and you'll notice how they went to different islands and became new species on these islands because they were separated for long periods of time. And so this is just showing you how from this ancestral bird it produces all these different species. And another example is like the Australian marsupials. And these marsupials, like we said, are mammals, okay? So they are similar to us. However, these mammals, when they end up reproducing and having their young, they have a different type of embryological development that happens outside of their uterus. And that's why they have the pouches like Kinga has right here. So at some point in time, they evolved from the placental mammals that we are, and it's due to an isolation. So most of these animals, these marsupial mammals are from Australia, and because they were isolated at some point in time by themselves, and enough to make them different. Placental animals, like is different from the marsupial ones because these are formed inside the uterus, and we complete that embryological development. And you see see the little giraffe coming out of the mother there. Yet another evidence for evolution is comparative anatomy, and like the name implies, we're comparing different species, their anatomy with other species' anatomy. And more pertinent to evidence is this homology that we see. And homology is the similarity that we actually see between structures within the species. So as you can see within the 4 limbs of animals here, our human, cat, whale, and bat, even though we have different functions for each of these 4 limbs of ours, however, structurally, we're really similar just said. And then there's also vestigial structures. And vestigial structures, these are structures that do not pose the same function or use as it once did from an ancestral organism. So, for example, they give an example of that ancestral whale, how now the whales, they don't have these particular bones, but this ancestral whale had these bones at one time, but they don't really have a use for it because it's not weight-bearing limbs for themselves. So it doesn't really have that function to bear its weight, so it doesn't really have a function. It doesn't share the same function as it once did when there was a four-legged animal. Another one is that some snakes have the pelvic and leg bones that you see over here. Not all snakes have it, but a few snakes do. And for us, we use it to help stabilize our lower limbs. But for the snakes, they don't have a use for it. So these are the vestigial structures. So they don't have a function for it anymore. And I remember Jackie was talking about how the appendix, right? Or not Jackie, Gina was talking about the appendix in a previous lecture. How that doesn't serve the same function as in other animals. Usually, other animals, they use that appendix to have a lot of bacteria in there to help digest the cellulose that they eat. So from grass or whatnot, from any type of plants. And we don't have that function anymore. Our function has changed. It's what's its function? Anyone remember?
Doesn't it help like the small or the large intestine with the immune system, right?
Exactly. It helps the immunity for our large intestine. Contains a lot of white blood cells and whatnot in there. And so it has a different function from what it used to be, but it's still considered a vestigial structure just because it's not the same function as it once was. Another evidence for evolution is comparative embryology. And like the name implies, you're just comparing the embryological development of different species. And as you can notice right now, like even a chicken embryo shown here and a human embryo, they have very similar structures, right? I mean, they look kind of similar. So both of them have these pharyngeal pouches. And what is a pharynx? Pharynx again? Yeah, it's that a region behind the nasal and oral cavity that goes down to the larynx area. And so yes, so this is pharyngeal pouches. So these pouches are found in that particular location. So as shown over here. So fishes, these pharyngeal pouches will end up forming gills. You know, I was trying to find ones for the other fishes in Finding Nemo. They don't have gills for some reason, so it was kind of interesting. For humans, on the other hand, those pharyngeal pouches will form the ear and also the throat that we talked about, just the pharynx itself. Finally, we have molecular biology that helps support evolution. One is the use of DNA sequences. And we use the DNA sequences of different species to see how closely related we are with those species. So how much DNA sequence match up with another species' DNA sequence. And if we're looking at the DNA of chimpanzee, you'll notice that humans share very similar DNA sequences with the chimpanzee, even more so than a gorilla, per se. So those that have more similar DNA sequences with each other are more closely related or have a more common ancestor that they have together. Questions so far? Can anyone name those 5 again? Things that help support or have evidence for evolution? Fossil record, biogeography, comparative anatomy, comparative embryology, and molecular biology. Very good. Okay, natural selection, once again, it's this process of which an organism, which tends to have beneficial traits that will be able to survive and reproduce and pass those beneficial traits to their next generation. So one thing about natural selection, first of all, usually when animals give birth, not all of them, but most will end up overproducing the amount of offsprings that they have. And by overproducing, it helps them for being able to carry on their genes for the next generation afterwards, because they will either die off by being hunted or by the environment itself. And not only that, sometimes by having this overpopulation, we end up having competition between each other as well. So there will be a limited amount of resources and space for these organisms to live in, and a limited amount of mates as well. So then they'll have to compete against each other. So there will be this trade-off between having the amount of offsprings you have so that they could have eventually someone to survive, versus to be able to have too many surviving and competing against each other. And those that have more beneficial traits will tend to survive and reproduce more so than those that have less beneficial traits. The other thing is that there is variation that will occur within each species. So each individual will tend to very different or have different types of traits that they have. As you can see here, all these ladybugs are slightly different from each other. They look similar, but by either the thickness of their spots or the amount of their spots, they kind of differ slightly from each other. And variation is very important, especially when we're talking about some kind of environmental change or whatnot. It may help these variations. Someone hopefully will have traits that will be able to benefit in that particular environment, so that it can carry on that species. So those that have more beneficial traits, like I've been saying, will be able to be more able to survive and reproduce successfully and pass on those traits to the next generation. So here's the Galapagos finches, and these Galapagos finches were found, or not found, but were studied by Darwin. And you'll notice how each finch, their beaks are slightly different from each other. So each beak here, like this one, tends to be a larger beak. And if it's a larger beak, it tends to be able to break down larger nuts. This beak is slightly smaller. It's more designed to capture insects. And as you can see, what this finch is doing is using their beak as a tool to be able to dig out little insects from the holes within this branch here. So they have different functions for these Galapagos finches, and it helps benefit each other, because if all of them have a large beak, they will be competing against the same resources, right? After the same large seeds and whatnot. If they have different beaks, they could coexist with each other. Okay, there are certain points that I want to talk about regarding to natural selection. First of all, natural selection is that the individual themselves do not evolve. What evolves again? It's the it's not the individual that evolves, but the species more general than species, the population itself, right? Which is the species, so all the individuals in that particular species. The population is the thing that evolves. So not like Charmander here, it's not the individual organism, but the population itself. Okay, natural selection can either increase or decrease the amounts of traits that are heritable. First of all, when we're talking about traits that are acquired within a person's lifetime, these are not actually genes, so it's not actually genetics. So someone like Gaston here, who actually develops stronger muscles, that's not, he's not born with stronger, let me rephrase this. He may be predisposed to have genes to produce these type of muscles, but he's not born with these big muscles to begin with. So, and these big muscles that he has will not be able to be passed on to his next generation, kind of thing. His potential to be able to develop bigger muscles can be able to be passed on, but the big muscles itself are not passed on. So that's what it means, that these traits acquired through life, if they're not part of their genetics, then it's not going to be passed on to the next generation. The other thing is that evolution is not directed to a certain goal. It's not trying to make a perfect organism or a perfect population, to be more exact, but more that it's using natural selection to kind of see which one's more beneficial for that particular environment. Like we were talking about between the white and black moths that we saw earlier, it's not, we're not saying that, okay, we're going to make a particular moth that's going to be benefit it for its entire existence. It just depends on that environment. At one point in time, it was more beneficial to be a white moth in that type of environment, but when the environment changed, then it was more beneficial to be the black moth instead. Another example of this is that there was a period of time where we used DDT as a bug spray. So it's probably most of you guys don't even know what DDT is, but regardless, so DDT was a bug spray at one point in time, and so there were some mosquitoes that were resistant to DDT. However, it has a detrimental factor to that as well. So even though they were resistant to DDT, it also decreased their growth rate as well. And so when DDT wasn't used at that particular time, the ones that were resistant to DDT were actually not as beneficial, so they tend to not be able to pass on their genes or traits to the next generation. But when we started using DDT, and all the ones that don't have this DDT resistant gene, those end up dying off. So the amount of these individuals that have this gene tends to increase the amount of individuals to have it. And so this is just this ebb and flow that you see within evolution, where we're not going to a certain goal, but more it's just this factor of how the environment changes that helps natural selection prefer a particular trait over another type of thing. And more examples. So pesticide-resistant insects, similar to what we were talking about with DDT. So here we see that there's these different types of, I guess, roaches here, and these roaches are a little more resistant to the pesticides that is affecting these roaches. So the ones that survive are these ones that have this particular gene that are resistant to this pesticide. And so they are able to survive and reproduce and produce more of these bugs that have drug-resistant insects. Another more pertinent topic is those that are antibiotic-resistant bacteria. As we continue to streamline the use of antibacterial soap and just medication and whatnot, we are kind of weeding out those bacteria that are not don't have those drug-resistant genes. And so when we weed those out, then those that are surviving are resistant to it. And so that's why doctors always emphasize to, when you're taking antibiotics, to take it throughout the entire period of time so that all the bacteria can die off, instead of some bacteria who are resistant to it passing on those genes to either another bacteria and then increasing the amount of antibiotic-resistant bacteria, and so on and so forth. And then there's also drug-resistant viruses as well, like HIV, that tends to resist different types of drugs. Okay, evolution tree. This evolutionary tree is this pattern that we see on the descent of ancestral. Oh, sorry, did I go too fast? Yeah, viruses. An evolutionary tree is this pattern that we see with the descent of an ancestor. So those that are more related or closely related tends to have a more recent common ancestor than those that are not so much related as compared to here. They do have a common ancestor, but it's a more ancient ancestor as shown over here. They're not as closely related. You can categorize these by either using homologous structure and or genetics. So those, remember how we were talking about the chimpanzees being really similar to humans, our DNA sequences are very similar. So we have a common ancestor, more so than the chimpanzee, than the gorilla that we saw earlier. And of course, those that are supported by both fossil record, comparative anatomy, and also the DNA data will become more and more accurate when we're actually doing these evolutionary trees. So as we're incorporating more and more genetics now within evolution, this is called modern synthesis, when we're combining the concepts of both genetics and with evolution. So first of all, some terminologies that you need to know. Gene pool. And gene pool is all the alleles that are within a population. You might not know what alleles are, but we're just going to talk about it in this case here. Allele is a gene that is actually found at a particular site of a chromosome. A chromosome is our DNA that is condensed, and there are different sites or different places within the chromosomes that will have particular gene in it. So over here, as you can see, this gene is for purple flowers here, and over here is for white flowers. It's just showing you once again that allele is just a location on the chromosome for a particular gene. And like I didn't mention, but you always get a chromosome from your mom and one from your dad's side. So anyone know how many chromosomes that we have in our body? 23 pairs of chromosomes. So 23 of them came from our mom's side. The other 23 came from our father's side. So a total of how many then? 46 then. Yeah. So example, like we just talked about, the enzyme that breaks down the pesticides, those pesticide-resistant insects that we saw earlier, that's located on a particular gene. So that particular allele has that enzyme that breaks down, while another one does not have that to break it down. So, of course, the amount of those that have this particular allele will increase if pesticides are used, because they kill off all those that don't have that particular enzyme that breaks it down, that allele that breaks it down. And so if they don't have it, they're going to die off and not be able to reproduce and pass it on to their offspring. Okay, a lot more terminology. Genotype. This is the genetic makeup of an organism. So when we're talking about the genetic makeup, we're talking about the DNA. So for example, like these alleles that code for type A antigens are erythrocytes. Anybody remember what are antigens once again? Disease-causing? No. Antigens are like these proteins that are on our cells. if we're talking about erythrocytes, they're on our erythrocytes to help us recognize our own cells, to recognize our own cells, so our immune system will attack it. And then versus if there's like a bacteria or something, they'll have their antigens, but that will be a foreign antigen, so our immune system will attack that. Pathogen, yes, pathogen. Yeah, it does sound similar. So the type A antigens will give what blood type then? Type A, yeah, So those that have the alleles for it, their genetics for it, will produce the phenotype of it. So phenotype is this physical expression of those particular genes. So we haven't really talked about this, but DNA codes for our proteins, right? So DNA, after it gets transcribed and then translated into proteins, that's our physical expression of our genes. So if we have alleles, this is part of our DNA that has this type A antigen, the phenotype therefore are the proteins that is made that is on at the surface of the erythrocyte, and this what gives us that type A antigens. Things that have polygenetic inheritance, like the name implies here, this is has more than one gene. So many genes are involved in this particular case. So these are traits that are encoded by these several genes. So usually it continuously varies, meaning something like height, okay? Height has many different areas or many different genes that helps incorporate height. So we're not talking about just one location or one allele. We're talking about different locations, different on different genes as well, to help us have this height. And you'll notice how it's kind of continuous. Continuous meaning it has this kind of like a bell-shaped curve. How towards the extreme, either very short or very tall people, there's fewer of them, right? Well, majority of them are in this middle range here of height. So I guess what I'm trying to get at here is that there are some genes that are in just one location, and these one location will either give, just say like we said earlier, a red color flower or a white color flower, while something that has a polygenetic inheritance will not only be located on this particular chromosome here, but it might be on a different chromosome located over here, so on and so forth. So it has multiple locations that will cause this continuous variation. A dominant allele usually overwrites a recessive allele when we're talking about phenotypically, and we usually use a dominant allele as a capital letter. So, for example, when we're talking about these red wildflowers, we're going to use the capitalized R here. Recessive alleles are not usually expressed, so they're usually the dominant allele will express itself over the recessive allele, and this is usually a lowercase letter. So if we're talking about the white wildflowers here, this is a lowercase r. So remember, genotype, we're talking about the genetic makeup. When we're talking about the genetic makeup, we want to know what that particular trait will be. So if it's homozygous dominant, that's when both alleles are dominant. So in our example about having the capitalized R as being the dominant allele and the lowercase r as being the recessive allele, what would be our homozygous dominant genotype then? Exactly, capital R, capital R. And homozygous means it's the same. So when we're saying the same dominant, then we have two capital R's. Heterozygous, on the other hand, is when we have one dominant allele and the other one's a recessive allele. So what's the, if we're going by the same example, it's going to be capital R and lowercase r. Very good. This will be a heterozygous genotype. Finally, we have homozygous recessive. This is when both alleles are recessive, so small, right? Small, lowercase r's. So regarding to this genotype, this is, remember, our genes, right, are part of our DNA. The DNA will eventually get transcribed and translated into proteins, and once it becomes proteins, this is where it actually gets physically expressed. So when we're talking about the phenotypes here, those that have the capital R genotype will actually have the physical expression of the dominant allele. So we're going to see red wildflowers. When we're talking about the heterozygous genotype, what do you expect the phenotype to be then? That's the genotype. Capital R, lowercase r, right? That's the genotype. What would the phenotype be? It would be red or white? It would be red because the dominant allele codes is for the red expression. So very good. The heterozygous genotype will code for red wildflowers. And finally, we have the genotype, the homozygous recessive genotype. So which was what? Yeah, it's recessive. It's going to be white. So the genotype, what is the genotype again? Lowercase r, very good. But the phenotype is the white wildflower. So far, so good? Yeah? A little bit.
It's just confusing because they're kind of similar. Geno and pheno. So
Yes.
just the genetic.
The genetic, yeah.
Would be the physical.
Yeah, the physical expression of it, yeah. Okay, when we're actually looking at a gene pool, remember what is the gene pool again? It's all the alleles in that particular population, right? So if the gene pool only contains these two alleles, this dominant and recessive, then the probability to get one or the other is going to be 100%. So so usually they use this lowercase p to denote the frequency of that particular dominant allele. And lowercase q is usually denoted as the frequency of the recessive allele. Okay, so you get this equation of the frequency of the dominant allele added to the frequency of the lowercase or recessive allele will give us all the alleles within that gene pool. So, for example, if the probability of getting the capital R allele in our wildflower example is 80%, and you guys all know how to convert that into decimal, yeah? You just go over a couple times, right? 1, 2, and so you get 0.8. Okay, so what is the probability of getting the lowercase r allele, the recessive allele? 0.2. How did you get that?
1 minus 0.8.
Yeah, okay, so the p equals 0.8, and then in this particular equation here, we have 0.8 plus q equals 1, and you just carry over the 0.8 over there, so q equals 1 minus 0.8, which equals 0.2. So what is the probability of getting it? How much percentage? 20%. Very good. That's a good question. Partially usually by looking at the phenotypes of how often it occurs across the population. Okay, so as an example, we could kind of see how many people are green-eyed in this classroom, right? However, that's only a phenotype, right? If it doesn't really show you the genotype of it, which we're using the alleles for here, right? Because someone with green eye color might be heterozygous, for example, and that's a different frequency of alleles compared to someone who is homozygous, right, dominant or something like that, and so we'll have to actually look at each individual of our DNA and look at those locations on our chromosomes that code for our green eye phenotypes, kind of thing. Does that make sense? Okay. Okay. Yeah, that's how we calculate it. Okay. The Hardy-Weinberg formula is this thing here. Is p squared plus 2 times p times q plus q squared equals 1. So p squared is this probability of obtaining a homozygous dominant genotype. Okay, so how do we get this? Well, we already talked about how p itself is the frequency of a dominant allele. So we also know that we'll always get an allele either from our mom's side or from our dad's side, right? So if we're getting an allele from our mom's side that's dominant, that's we could say it's this frequency of us getting a dominant allele on our mom's side, and then we have to multiply that by that frequency of an allele on our dad's side. So that's how we get this p squared. So you're basically multiplying the frequency on the chromosome that you obtained from your mom's side, and the allele frequency that is obtained from your dad's side. So that's why you multiply by themselves, and then you get this p squared here. When you have this 2pq, this is this heterozygous probability. This is due to a couple of factors. One is that when this is what is this again? Homozygous dominant of a genotype, right? Here we have heterozygous genotype. Very good. However, you'll notice that there's two ways of combining this, right? If we got this from our, just say, mom's side, and we got this from our dad's side, right? Even if you swapped it around, it's still the same thing, right? If we got this dominant allele from our mom's side, but this smaller recessive allele from our dad's side, there is a potential we could also get the recessive allele from our mom's side, right? And then our dominant allele from our dad's side, right? And so that's why there's this 2 times. So you're multiplying it by 2. So you're multiplying by the frequency of the dominant times the frequency of the recessive, and you multiply by 2 because it could be swapped around.
Are we allowed to write it that way?
Say again? Oh, this is just telling you the frequency of it. We're not talking about the alleles itself. Just how frequent, or what's the probability of getting that particular dominant or recessive allele. Yes, PQ twice, and that's why we get this 2 here. Yeah, that's okay. And then there's this probability of getting homozygous recessive, right? The smaller case RR, right? So this is the probability of it. Same thing as how we calculate the probability of the dominant genotype. So going back to our example, if the probability of getting a large or dominant allele for red wildflower is 80%, then we already calculated the... Hey. Yeah, no problem. Okay. So using this example, going back to the example, so if 80% is for the dominant allele, we already calculated what's the recessive allele frequency? 20%. Okay, good. So using those probabilities, now find out the homozygous dominant genotype probability of getting that, and the heterozygous, and also the homozygous recessive genotype as well. So do that now. Okay, so once again, the probability of getting the dominant allele is 0.8. This is the probability of getting this dominant allele. Probability of getting a recessive allele is 0.2. This is the, sorry, it's a lowercase r here. So what's the percentage to get a homozygous dominant? This is the genotype once again. It's basically p squared, which is 0.8 times 0.8, which is 0.64, which is 64%. Okay, so what is heterozygous? How about just getting this heterozygous genotype? Okay, and how did you get that?
2PQ is the formula. So
Okay.
It's 0.8
0.8
times 0.2
times 0.2 and
0.32.
0.32. Okay, very good. So, no, that's not 0.16. Yeah. So, yeah, but getting just this genotype is only 16%. If we were trying to get this heterozygous genotype, right, it's still the same, right? And that's why I was talking about how that 2 came into play. This 2 came into play because either you could get this on your mom's side, right, and get this on your dad's side, but you could also get this on your mom's side and get this on your dad's side, you know, kind of thing. And that's why you multiply it by 2. So in this case, we're going to add it. But I mean, if you multiplied any one of these by 2, you would still get this 0.32, which is 32%. That's to get the total heterozygous genotype. Yeah, not just one type of heterozygous genotype or the other. No, I'm just kind of clarifying, like, do you understand the concepts behind this, you know, kind of thing? Instead of just plugging in the equations, like, okay, what does that mean, though? Yeah, kind of thing. Okay, so, and finally, what's the recessive genotype? Yes, lowercase r is the genotype. And the probability of getting this genotype is 4%. And you got that by, yeah, the q squared, right, which equals 0.2 squared, which equals 0.04, which equals 4%. And you'll notice if you want to just double check yourself and see whether or not you did this correctly, just add them all up. So here you have 64 added by 32, that's 96. And add by this 4, and you have 100%. Hopefully your score at the end of this test, you know, kind of thing. So that's how you kind of determine it. So I kind of rewrote it out here, but so on and so forth. Finally, the thing about Hardy-Weinberg, this formula only occurs in a hypothetical situation or an ideal situation. It's when we reach this equilibrium called a Hardy-Weinberg equilibrium. This is when the population has a genetic equilibrium. So the frequency of the dominant allele does not change, and the frequency of the recessive allele does not change. And so, of course, if it doesn't change, this particular population, if it's not changing, is not evolving. Does that make sense? So in order for evolution to occur, natural selection will be in place. So certain traits are favored over another. So you'll end up varying the amount of frequency of these alleles. Right? And if you're varying it, then you can't really use these equations because they're constantly changing. But we're using this Hardy-Weinberg formula as like a hypothetical situation, an ideal situation where there is no changes are involved. So we have this genetic equilibrium. There's no change occurring. Another way to kind of help you out with this probability is using this thing called the Punnett square. And a Punnett square does help somewhat if you were to draw a square out, and then you put the alleles on the male or female side, and then you'll end up finding the probability of that particular generation that they end up producing. So I'm going to just recreate this and make it a little more simpler for you guys to understand here. So if we have a heterozygous mother, then she'll have these alleles here, right? If we have a heterozygous father, they'll have what will it be over here? Big R, and this one will be little r. Okay, so both parents are heterozygous. And so if we look at this here, this offspring will be homozygous dominant. Very good. And this offspring here? Heterozygous, right? So what I did was just put the R here and another, this capital R here, this capital R here. This case scenario, I put the capital R here and the lowercase r here. How about this one? What is this going to be? Yes, heterozygous. It's the other way around, right? Where we have this lowercase r here and this capital R here. But, I mean, it's the same, though. It's just the alleles are coming from different parents. So I'm going to just rewrite it into the normal way that we normally write it. And over here will be what? Homozygous recessive. Very good. Okay, and you'll notice that the probability now of children will have a different ratio, right? So how many children will have a genotype of... Sorry, what's the probability of having a child that has the genotype of a homozygous dominant? 1 out of 4, right? 1 out of 4 children, 25% of the children, right? Because there's 1 out of the 4 genotypical children that you'll have. How about for heterozygous? 50%, right? A quarter over here, another quarter over here. Add them together, it's half. So on and so forth. Okay. So let's go. Okay, why don't we say it's a homozygous mother, homozygous dominant mother, and homozygous dominant mother mated with a homozygous recessive father. Okay. So homozygous dominant mother and homozygous recessive father. By the way, hopefully you guys know male and female like symbols. I use that quite often. So this little symbol here is for females. This symbol here is for males. Okay, so homozygous dominant, first of all, what's the genotype? Capital R, very good. How about homozygous recessive? Lowercase, very good. And if we draw a Punnett square here. Okay, this child here will be what genotype? Heterozygous, very good. This one is also heterozygous. Yeah, very good. They're going to all be heterozygous. So what's the percentage of any heterozygous child? It would be 100%. Okay.
100% of the time we'll get that trait.
Yes, 100% of the time this 100% of the time their child will have this particular trait. Yes, very good. That would depend. It really depends on the genetics. And some of them, like when we're talking about blood type, is codominant, meaning that blood type A, blood type B, and blood type AB are all dominant. It's O that's recessive. Okay. And you can actually calculate that out too if you are to have your parents' blood type. Like for example, my parents' blood type, one is heterozygous A, the other is heterozygous B. Okay. So, what will this particular genotype be? Yeah, it will be AB. Blood type AB. How about this genotype here? Okay, it will be a heterozygous, right? Of this immunoglobulin B. Okay, how about this one here? Immunoglobulin A and lowercase I. This one here. 2 lowercase i's. So, phenotypically, so this is the genotype. Phenotypically, what is this? Type blood AB. Very good. What phenotypically is this? Type B blood. Very good. Phenotypically, what is this? Type A. And phenotypically, O. Very good. This is type O blood. So you can actually calculate, your percentage of your children to be what blood type and stuff. Of course, you'll need to know a little bit about your genetics kind of thing. The reason why I know my parents are heterozygous A, heterozygous B, is because I'm a blood type O child. So it must, unless something happened that I did not know about kind of thing. But which does occur actually. Go ahead.
Would that be out of 4 kids, at least one will come out? Oh.
Yes. I mean, probability-wise, right? 1 out of 4 children, probability-wise. So each child will have a 25% chance of getting blood type O. Okay. So we were talking about, so these are the genotypes, right? So phenotypically, what is the percentage of getting red wildflower? 75% phenotypically. Very good. And white wildflower? 25. Okay. A little harder question here. So people who have this phenylketonuria, PKU, cannot break down this amino acid called phenylalanine. So this may end up causing mental retardation. So PKU actually has a frequency of having 1 out of 10,000 babies born in the U.S. and is also a recessive allele. Can you actually calculate for me the frequency of this allele and also the frequency of the genotypes of the homozygous dominant, heterozygous, and homozygous recessive? Try that out. So I was saying in class how this 1 out of 10,000 babies were, is this a phenotype or genotype? And we discussed this and we concluded that it's a phenotype of what's being expressed. And so if that's the phenotype, we know that from the Heidi-Weinberg equation, to be able to have a phenotype, we'll have to have a homozygous recessive for this particular disease. And to calculate a homozygous recessive, it's this Q squared. So we're going to make that equate to 1 over 10,000. And so we'll end up square rooting each side here. So that will be Q equals what then? 1 over 100, very good, which is 0.01. So this is 1%. For homozygous recessive, very good. So, what is P then? 99%, right? It's just 100% or 1 minus 0.01, which is 0.99, which is 99%. Okay, that's really messy. 99%. Okay, so knowing that, okay, so what's the probability of gaining a homozygous dominant genotype?
That's the 99%.
Yeah, it's,
That piece over p squared is what you're asking.
yeah. So p squared equals 0.99 times 0.99, which 98, right? Okay, which is 98%. Okay, and the frequency of getting heterozygous 2 times PQ, right? So it's 2 times 0.99 times 0.01, which equals 0. What? Sorry? 198.
Excuse me, 0198.
Okay, that's what, yeah. 0198. So it's basically what?
2%.
1.98%. Okay, so we already determined what is Q squared again? It's this, right? 0.
0001.
Something like that. Okay, so it's very infrequent for someone to is born with this, but it's frequent enough to be able to actually have these printed out on soda cans and whatnot that uses this particular amino acid. Okay. There are different sources of genetic variation. One is through mutations. And mutations is actually changes within the DNA itself. So the DNA has a sequence, and you'll end up changing the sequence, some nucleotide or something like that, that will end up affecting the expression of DNA. So usually, most of this is detrimental, most of the time. So it's very few times that we'll actually have some sort of superpower or something beneficial in that particular instance. But it does occur, right? I mean, we talked about how there's pesticide-resistant bacteria and insects and stuff like that. Sorry, pesticide-resistant insects, but antibiotic-resistant bacteria and things like that. So it does occur. And the thing about mutation, it increases the genetic variation. And remember, variation is always a good thing because in case of some kind of environmental change, that particular organism may be able to survive. So usually we end up increasing the amount of variation with organisms that have shorter life periods. That way they could end up reproducing more and more of this particular mutated trait. Sexual reproduction, they have different ways of increasing the variation. One is through crossing over, another is through random assortment, and also the randomization of fertilizing just random eggs and sperm also increases that variation as well. So crossing over, this actually occurs in prophase 1. We haven't talked about meiosis yet, so just keep this in the back of your mind, okay? So prophase 1, what happens is that these homologous chromosomes get really close to each other. The homologous chromosomes, and once again, one is from your dad's side and one's from your mom's side. They get really close to each other that they actually swap parts of their chromosomes with each other. And you'll notice how they come out with something different. That makes us not identical either from our mom's or dad's side, or even from our brothers or sisters as well. Random assortment also occurs, and this happens in metaphase 1 or anaphase 1, one or the other. And this is when they actually kind of line up in the middle here. And you'll notice how when this lines up here, when it lines up here, they get assorted. So on this particular eventual sperm or egg, this ended up getting carried over to this cell here, which end up dividing to form these particular gametes. Gametes are sex cells, which is the sperm or egg. And on this particular site here, after meiosis, ended up having, just say both from the mom's side, and producing these sex cells here. But notice how we could say that it got assorted differently here. Notice how this here, we got one from our dad, and one from our mom's side. And you'll notice how this is actually ends up producing different types of gametes compared to these ones here. You guys see the difference between the two? No? Yes? And this is this random assortment. Like, we randomly end up, or our cells randomly ends up sorting it out so that we end up getting different types of alleles at the end. And this was randomly assorted so that we end up getting these alleles at the very end. Okay, that's random assortment. And this occurs during this metaphase or anaphase 1.
Is it? I don't know how to form my questions. Is it that we don't know in which phase it occurs, or is it just?
Oh, no. It's not that we don't know which phase. It's just some people say it's metaphase that it gets determined. Other people will think that, well, at metaphase they just line up there. In anaphase they're actually pulling the chromosomes away. So it really just depends on your perspective on seeing things. Yeah. Finally, we come with random fertilization. So we could have certain sperms will fertilize certain eggs and so on and so forth. So when we're talking about these sperm and egg, we could say that this sperm here fertilizes this egg, or we could say this sperm here fertilizes the same egg, and so on and so forth. And we just end up, notice there's so many different combinations, right? This is if both of them fertilize that particular one, but this is a different type of egg, right? So if we fertilize this is a different combination of the chromosomes. So it's just showing you that all these produces more variation for our offspring, so that they could potentially have better traits for a certain type of environment. Okay, so there are mechanisms for evolution. One is this genetic drift, and this is when there's some sort of change within the gene pool of that population. It's usually due by some sort of chance. This is very dependent on sample size. What is sample size? This is the amount of individuals or in that particular population. So when you have more and more individuals, then you have a smaller chance of affecting this genetic drift, because you have a large population and any kind of change, genetic change within it, is not going to affect it drastically. However, if you have a very small sample size, so very few individuals in this particular population, then if you end up changing the gene pool in there, somehow it's going to cause a drastic change within that gene pool. So, for example, over here, normally, as you can see, the frequency of the recessive allele is only 30% here, while the frequency of the dominant allele is 70% here. If we only take these 5 individuals and just end up reproducing them, we end up decreasing the sample size, right? We only chose 5. We're not having all of them reproduce. We're just choosing 5 of them. And when they reproduce, you'll notice how the frequency of the recessive allele now has increased. If we end up even decreasing it even more, the sample size, just choosing these ones here, we'll notice how the frequency of the recessive alleles has disappeared. So this is just showing you this genetic drift happening, where the smaller the sample size, the more genetic drift that there is. There's more changes within the gene pool itself. Okay, so that's why it's so important when we're talking about things that are endangered species or species that are becoming more and more extinct, because there's not that many of those particular individuals, and if they have some sort of genetic defect, then potentially everyone in that future generation will have that potential genetic defect as well, because it's a smaller sample size, and this genetic drift could potentially occur. That smaller sample size will have those particular traits from those individuals, and it might not be a representation of when it was when the endangered species were not endangered at that point in time. There's different types of genetic drift, bottlenecked and founders, and we'll go into that. And then besides chance with genetic drift, there's this thing called gene flow. And this is when population either gains or removes a particular allele due to individuals that migrate. So it's not really by chance. It's actually there's it's not just by chance. So for example, like these plants over here, they are able to potentially transfer some pollen or whatnot onto the other side of this lake here or river, so that you end up increasing the amount of variation. So bottleneck, this is this genetic drift, like we said. So it ends up drastically decreasing the amount of population size that there is, kind of like if you were just pouring these beads out of a bottle. So usually this is caused by some sort of environmental changes, either through earthquakes, floods, or fires, and that will end up causing a smaller sample size that end up surviving these natural disasters. Or unnatural in that case, but anyway. So here, so there's a cause of the causes of decrease in variation is due to some sort of alleles that are lost through the gene pool due to the environmental changes. And if we end up decreasing the variation, then it's going to be representative for that particular new population. So it will end up also decreasing the chances for to be able to adapt to new environmental changes. Does this make sense? Like, when a population, a lot of them end up dying, you only have a smaller sample of it, then whatever gene pool that once was will be limited to now the gene pool of these individuals there. So you'll have a smaller, you don't have as much variation anymore as you once were. Founder effect is kind of similar, but it's not due to some natural disaster. It's when you actually have a genetic drift when a population ends up establishing itself in a new environment. So this is what happens actually when we're talking about the finches and organisms on the Galapagos Island. The Galapagos Islands are very isolated, or any kind of islands are isolated, and over time, if they get separated from the other same species on another island, they might end up having particular traits that they develop more specific for that particular island, and then become a separate species in itself. Not a different race, because we actually are not a different species. We can still reproduce, have fertile offsprings, kind of thing. But for example, I'm going to just give this as a crude example. If this particular finch was on this particular island, and it cannot reproduce with another finch on this particular island, just say, okay? If they try to reproduce, it may have an offspring, but that offspring may not be fertile. So, for example, like if you mate a horse and a donkey together, you'll get a mule, but the mule itself is not fertile. It cannot produce other mules. Kind of thing. So that's why they're different species. So horses are a different species than a donkey. Okay, once again, gene flow is this migration of alleles where you have one population here. So in this particular population, it is against the recessive alleles or recessive phenotype, while this population here tends to not have any dominant alleles in this particular case. However, you'll have some that ends up migrating over to the other population, and by migrating to the other population, it introduces these alleles in that population. And by introducing those alleles, you end up increasing the amount of genetic diversity as well. So it increases the variation. Okay, so in this case, they're gaining these alleles by migrating over. And like I said, it helps reduce the differences between the populations. So in this particular case, even though they are separated by that mountain, they might still be the same species, but even though they're separated. Okay. Genetic drift, gene flow, and mutations all increase the amount of variations that occurs within the gene pool. And it does help the population to potentially be fit for an environment that might occur. However, it does not cause natural selection. Remember how we talked about natural selection? It promotes the adaptability for that particular population to eventually evolve. And this is due to fitness. And fitness is what an individual can contribute to that gene pool. So usually if that individual survives and reproduces, it then contributes its traits to that gene pool so that the next generation can have those traits. So in this particular case, the more fit is the black moth here to reproduce and cause those genes and traits to be passed on to future generation. However, the white moths, if they end up dying and not being able to reproduce, then they're not able to pass it on. So they'll be less fit. So there is different types or modes of natural selection. One is directional selection. Directional selection is when there's this shift in the population. So here's the original population here. And you'll notice that there's a drastic variation of the different types of phenotypes of this particular rat or mouse here, from white fur all the way to dark fur. And when one is naturally selected over another, these are more preferably selected, then it ends up eliminating the extremes here. So there's less so here. And this directional selection shifts this curve to the right so that it tends to have more of the darker fur ones while less of the lighter ones. And once again, this usually occurs with some kind of environmental change that will occur, or when an organism ends up migrating to a new environment. And we gave an example of this, how pesticide-resistant insects shows this directional selection. You'll notice how these have different genes. Some are more resistant to the pesticides, some aren't. When you use the pesticides, the ones that are resistant survive, and only the ones that end up having this resistant gene will be able to have that continue that population. So we end up not selecting those that were not resistant to that pesticide. Disruptive selection is when you end up increasing the contrasting phenotypes in that population. So you either select the very lighter colors or very darker fur colors of the rats, but none of the intermediates. Usually this occurs when you have patchy environments that either favors one or the other type of phenotypes, but not the ones in between. An example of this are the garter snakes. The garter snakes here, some are striped as this one here, and some are spotted. Spotted. And they tend to are more selected more than those that are kind of in the intermediate. The ones that are spotted tends to have a behavior where they just stand still and kind of camouflage with the environment. Those with the stripes tend to slither away, and due to it slithering away and the stripes causes confusion or confusion of whatever predator might be attacking it. And so so they tend to survive more so than those that are not spotted or striped. Stabilizing selection favors the intermediate phenotype, so it's the antithesis of the disruptive selection. And this occurs usually in stable environments. An example of this, so it selects against the extreme phenotypes, so it's the opposite of the disruptive selection. Example of this are baby weights, and this tends to occur most of the time. So weight of babies, right? Babies that are way too small or way too big tend not to survive, but those that are in the intermediates tend to survive. Those that are too small may not be developed enough, and so they die. Or those are too big that could be potentially so big that it doesn't pass through the birth canal of the mom, causes death of the baby as well. So those that are intermediate tends to survive. Finally, we have sexual selection. And this is a type of natural selection that ends up having the individuals with certain traits preferring other mates, and it increases the amount of chances of passing down those traits. So we have males and females, of course, and there's this thing called sexual dimorphism. It's more apparent in animals than in humans, but there's this distinct appearances between the two sexes, not only when we're talking about genitalia and stuff, but more about other outer appearances as well. So males tend to have more vibrant color or adornments. The adornments like the antlers or manes or feathers. So the thing about this is that these adornments may help compete with other males for potential mates. For those vibrant color and things like that, it may signify that those particular males are healthier and so have more beneficial traits for the females. And of course, if they're competing against each other, then they're stronger, and so the ones that win are stronger and so have better traits that the females may prefer. While females, on the other hand, tend to have less colorful, and it's due to evolutionary reasons. If they're less colorful, they're not as potential targets for predators, right? And so that they could end up protecting their young if they're raising their young. And they also have a preference of choosing healthy males. They end up choosing who they want to mate with. And they do that by, once again, by how the males present themselves.
So, like for the peacock,
Yeah.
would it be the female going for the male that has a brighter color?
The brighter color with a lot of feathers, and you see them fan out and dance and stuff like that. That's the males. Yeah, those are the males.
Males are looking for those that have like the fuller feathers and.
Yeah, because if they have fuller feathers and more colorful feathers and things like that, it kind of signifies for the females that these that have more vibrant colors and stuff have good genes in them potentially, And so,
Does that not, like, the vibrant colors and stuff, does that not pass on to their offspring?
that does.
It's a female?
Oh, those genes are not coded for the females themselves. I mean, they do pass it on to their offsprings, yes, but they're not expressed though. Uh-huh. Good. Yeah. Oops. So I think acknowledgements, pictures, and I'm going to stop.